Hello, everybody. I'm Lino Nobili from the University of Genoa. Very happy to be here, to be involved in this workshop. Thank you, Liborio and Ivana, for this kind of invitation. The title of my presentation is REM sleep and epilepsy. I don't have any conflict of interest with respect to this talk. These are the outlines of my talk. We talk about the relationship between REM, seizure, and epileptic discharges, the role of REM sleep in lateralizing and localizing the epileptogenic zone, the suppressive effect of phasic REM sleep, the effect of seizure and epilepsy on REM sleep, and then we give you some insight regarding REM sleep and epilepsy treatment. So, first of all, we have to say that epilepsy is a very common neurological disorder. Around 50 million people worldwide have epilepsy, and 1/3 of patients are drug resistant, so they do not respond to treatment. Let's now move to the role of REM sleep in preventing both seizure and epileptic discharges. The high majority of studies have been conducted in the past in order to investigate the role of non-REM sleep in activating spike and epileptic discharges. However, I think that it is extremely important and curious also that REM sleep is really a strong suppressor, exerts really a strong inhibition on epileptic discharges and seizure. This has been confirmed by many studies. In this review by Marcus and Pavlova, it has been shown that only 1% of seizure arise from REM sleep. And also interictal discharge rate is low during REM sleep. So REM sleep is the most protective stage of sleep against focal seizure, generalized seizure, and also focal interactive discharges. Not only spikes, but also REM sleep is able to reduce fast ripples, HFO, high frequency oscillation, that are a strong biomarker of epilepsy, as shown in this publication by the Canadian group. But REM sleep can also lateralize and localize the epileptogenic zone. So interictal epileptic discharge in REM sleep can be useful for lateralizing, localizing the epileptogenic zone. Again, in this review, Marcus and collaborators have shown that spikes occurring during REM sleep correctly localize the epileptogenic zone in 84% of cases. And the REM spikes were never false localizing. Indeed, this is a topic that still is a matter of debate because there are some studies suggesting that indeed could be also REM sleep, the state of vigilance that best identifies the epileptogenic zone. These are studies conducted with machine learning techniques, and we will see in the future. However, it's true that although spikes are rare in REM sleep, when they occur in REM sleep, they generally are localized inside the seizure onset zone. But let's now move to the topic of most important aspect of my talk. So the suppressive role of phasic REM sleep on epileptic activity. We know that REM sleep is not an homogeneous state. It's characterized by two completely different states. Phasic REM sleep with bursts of ocular movements, muscular twitches, and tonic REM sleep, where there is the absence of ocular movements, although there is the persistence of muscle atonia. And these are two completely different neural states. Indeed, during phasic REM sleep, there is a extreme internal activation of the cortical and subcortical networks. There is an activation of the motor cortex. There is also an alteration, a modification of the synchronization inside and between the hemispheres And during this activation, however, the brain is less able to integrate information from the external world. There is also an activation of the limbic system, an activation of also the thalamus and subcortical regions. So completely two different states. REM sleep during the phasic activity is very similar to a state of active wakefulness. So the motor cortex is activated like during movements, for example. And also the occipital region, the visual system, is extremely activated. So, which are the effects of phasic REM sleep on epileptic activity? It has been shown using SCAL-PG that there is an inverse relationship between the number of ocular movements and the number of spikes. So the more we have ocular movements during a REM episode, less is the production of spikes. And then in another study using foramen ovale electrodes, it's been shown that the reduction of spikes at the level of the temporal lobe is extremely evident during phasic REM sleep with respect to tonic REM sleep. More recently, and this is studied again from the group from the McGill University, Birgit Frauscher and Professor Gottman, they showed that during phasic REM sleep, there is not only a reduction in spikes, but also a reduction in pathological high-frequency oscillations, so-called fast ripples. These are particularly reduced during phasic REM sleep with respect to tonic REM sleep, and obviously also with respect to non-REM sleep. Similar study was conducted by our group in two different types of epilepsy, subject with focal cortical dysplasia and subject with hippocampal sclerosis. We investigated the production of interictal discharges at the level of the seizure onset zone and at the level of the diffusion zone, so where the ictal discharge spreads. And we observed, in parallel with the study of Birgit Frauscher, that the spike rate was notably reduced during phasic REM sleep, both in patients with hippocampal sclerosis and focal cortical dysplasia, and both in the seizure onset zone and the diffusion zone. Such a reduction was statistically different with respect to tonic REM sleep and with respect to non-REM sleep. And here you have an example. You see here an electrodes recording from the focal cortical dysplasia inside the seizure onset zone, and here electrodes recording from the diffusion zone. During burst of ocular movements and during REM sleep, you can notice that there is a reduction in the amplitude of the spikes at the level of the seizure onset zone, and there is also a reduction in the spread of epileptic activity outside the seizure onset zone, while during tonic REM sleep, you can notice that spikes has a higher amplitude and spreads also in the diffusion zone. And the reduction of spikes during phasic REM sleep was also demonstrated in these peculiar epileptic syndromes, electrical status epilepticus during sleep, that is an epileptic syndrome that can appear in children. Here we have recently demonstrated that the spike index in phasic REM sleep was reduced by 87% in phasic REM sleep with respect to tonic REM sleep. So phasic REM sleep is able to reduce spikes also in an extreme condition like electrical status epilepticus during REM sleep. What about the possible physiopathological mechanism aspects related to the suppressive role of REM sleep, phasic REM sleep on epileptic activity? Well, we know that while during non-REM sleep, there is a modification in the firing pattern of neurons. They enter into so-called bistable state characterized by up and down state. That this is a state that is well known to favor seizure together with arousal fluctuation. During REM sleep, there is a tonic firing on neurons similar to the one of wakefulness, and there is a production of acetylcholine like during wakefulness. And in this important study conducted by Professor Schaus many years ago, it has been shown that if we remove the desynchronization operating during REM sleep, we can have epileptic discharges and seizure also during REM sleep. And this can be done, such a kind of dissociation can be done using atropine. Atropine can create a slow wave sleep-like EEG synchrony in REM sleep, and selectively such dissociation can increase the incidence of epileptiform discharges. In an animal model of epilepsy, it has been shown that such dissociation, so the production of synchronization during REM sleep, can favor the occurrence of spike and interictal discharges. So it seems that acetylcholine is really probably the principal cause of such a desynchronization and such an effect on epileptic activity. However, in another study, it has been shown that the production of PGO waves, pontogeniculocippital waves that are typical of REM sleep and of phasic REM sleep, PGO waves, independently if they are triggered during REM or during wake state or during non-REM, they can exert an inhibitory influence over the number and duration of seizure, over spike frequency, and they can even restore sleep alteration produced by experimental epilepsy. And moreover, it seems that during the production of PGO waves, there is a further increase in cholinergic activity. So we also measured the level of synchronization operating during phasic and tonic REM sleep. And what we have found is that during phasic REM sleep, there is a reduction in the global and local level of synchronization. So we can suggest that during phasic REM sleep, at the local level, at the level of the epileptogenic zone, there is a kind of a reduction in connectivity and synchronization inside the lesion, and also a reduction in connectivity and synchronization at the global level. While this is different outside REM sleep, at the local level, during tonic REM sleep, there is an increase probably of synchronization, and also an increase of synchronization outside the epileptogenic zone. Which are the effects of seizure and epilepsy on REM sleep? We know that seizures, also the seizure occurring during wake, can reduce the amount of REM sleep, and the seizure during sleep also can reduce the amount of REM sleep, especially if they occurred in the first part of the night. And this was one of the first studies on this topic. And more recently, it has been published, this review and meta-analysis confirming that seizure and epilepsies are associated with the decrease of REM sleep, both focal and generalized epilepsy, and independently from drug treatment. And such a decrease is especially evident in subjects affected by drug-resistant epilepsy. Indeed, in patients with drug-resistant epilepsy submitted to surgery and seizure-free after surgery, is clear, evident that there is a rebound of REM sleep after the intervention. So it seems that REM sleep, the reduction of REM sleep seems to be a kind of biomarker for epilepsy. In this retrospective case control study, it has been shown that the reduction of REM sleep is particularly evident in subjects with epilepsy with respect to subjects with acute symptomatic seizure or psychogenic non-epileptic seizure, syncope, and other neurological disorders. But there is not only a reduction in the total amount of REM sleep, there is also an alteration in the microstructure of REM sleep. So seizure and epilepsy seems to be also related to an alteration of microstructure of REM sleep, in particular in a reduction of sawtooth waves, especially in extratemporal lobe epilepsy, as recently shown by the group of Birgit Frasche. And I think it's interesting to underline that it has been also published, this paper some years ago, showing that there is a kind of atrophy of the pedunculopontine nucleus that is strongly involved in the production of REM sleep and also phasic event during sleep. There is a kind of atrophy in patients with sleep predominant seizure. So there is kind of really also a reduction in REM sleep, but probably also an alteration in the morphology and functional aspect related to the morphological and functional aspect related to REM sleep. So just to conclude, some observation on REM sleep and epilepsy treatment, only focusing on orexin. Indeed, we know that orexin is completely absent in REM sleep. And we know that in experimental model that the orexin receptor antagonist, almorexant, can reduce seizure, the incidence of seizure, can reduce the latency to REM sleep and reduce the seizure burden of epilepsy. And so, as also suggested in this review, in the future, it could be that the orexinergic modulation could be used to treat seizures and epilepsy. This is something that must be studied in the future. I would like also to underline the fact that in patients submitted to vagal nerve stimulation or deep brain stimulation, when subjects that are drug resistant, it has been shown, for example, in animal models that vagal nerve stimulation not only reduce seizure, but also increase REM sleep, and particularly increase the production of PGO waves and the number of ocular movements. The same line, in this more recent study, it has been shown that the deep brain stimulation not only reduced seizure, but also increased the amount of REM sleep. So it seems that increasing the REM sleep could be paralleled by a reduction in seizure. We don't know really the causality between the two phenomena, but it's been proposed, and with this we conclude that could be that the pedunculopontine nucleus that is involved in the production of REM sleep could be a potential novel deep brain stimulation target in subjects affected by drug-resistant epilepsy, and we need studies on this topic. So, to conclude, ictal and interictal epileptic activity are significantly inhibited by REM sleep. REM sleep seems to be to better localize the epileptogenic zone, although there are no definitive results on this topic. The highest inhibition of ictal and interictal epileptic activity occurs during phasic REM sleep. Increased epileptiform activity during REM sleep seems to be associated with increased seizure burden. Seizure, both during wake and sleep, reduce REM sleep. Increased REM sleep and lower PGO waves may reduce seizure, according to experimental investigation. And so, finally, understanding why REM sleep reduces epileptic activity may help develop new treatments for people with epilepsy. Thank you very much for your attention.